Welcome to this presentation. We are going to present S-Aligner, the software for genome assembly developed by Contignon Technologies. Genome assembly is a fundamental step in genome sequencing. There's no technology, actually, able to obtain the full DNA sequence of any biological entity in a single read. What you obtain, instead, are multiple reads of the sequence, and you need to combine these reads forming a larger one. This is like a gigantic puzzle and indeed a computationally hard problem for which, given its relevance, many different software solutions have been developed. S-Aligner is one of these solutions, and as we will see, the one actually obtaining better results. In these charts, we see some results obtained applying different software to some benchmark sets established by the community to evaluate the performance of genome assembly software. In the first case, we compare S-Aligner results for a dataset containing sequencing reads from different viruses. We see how S-Aligner outperforms on every case all the other ones, and on average it gets a 110% increased performance over the second best software. In the second chart, we see how it gets a 64% increased performance for a especially hard benchmark set containing large viruses from mixed strains, which is a circumstance usually found in viromix studies. Here we see some concrete results. We see how in every case S-Aligner gets sequences with almost double the length than any other software. In the case of the HIV virus, which is around 10,000 base pairs long, we obtain almost the full sequence, while other software obtain only half of the sequence in the best case. Even if we use less quality data, like in the third table, we obtain a better assembly than with other software. In that case, we obtain a 9,000 base pairs sequence using Roche data, while other software obtains 3,200 base pairs, in the best case, even using better sequencing technology. We have also made tests for the SARS-CoV-2 virus. We downloaded public datasets shared by scientists and evaluated S-Aligner performance compared to other software. As we see, using SPADES, the best of the other software, we get on average sequences with around 17,000 base pairs. In contrast, we see how S-Aligner almost in every case obtains the full sequence and on average 28,000 base pairs, which is almost the double than the second best software. Thanks for watching. Please do not hesitate to contact us if you have any doubt or if you want to test our software.